Intelligence. Wow, this is the first time I've been in such a conference about something as important as all of that. And most of you, I notice by looking at the program, are very much interested in this animal right at the end here. And we, of course, would like to know how our intelligence arose, but we might have to look back a very, very long way. Only in the last 500 million years or so were there any multicellular organisms. And for about 2 to 3 billion years before that, there were unicellular animals and organisms which had to live through a pretty tough time. If you thought Brexit was tough, look at this. The whole Earth was covered with snow and all sorts of tremendous things. The animals and organisms that lived back then were remarkable in their ability to do many very clever things, including finding lunch. So how does a bacteria find its lunch? Well, this is what they look like, and they can swim around the place in the soup that they live, and what they want to do is to find the food. And all food has a concentration gradient, and somehow this little organism has to swim up that gradient and find its way toward the food. Now, cleverly, about 4 billion years ago, was a very exciting little molecular invention made up of only 2 proteins that sits in the membrane of the bacteria, and it has a sensor for the chemicals that are emanating from this sandwich, and then it can use this to detect the fact that this is releasing the food. But there's a huge problem, and the huge problem is this, that at the level of the bacterium, where it has these little molecular machines on it, there is no gradient. It's too small, so it doesn't know which way to swim. But they solved that problem through molecular evolution because the bacterium invented the ability to measure time. And the way it does that is that it detects the signal at the first time and it senses it, and there is a molecular change here. And then it swims randomly, and then it takes another measurement. And it, by virtue of the timing, can detect if it is going up the gradient or down the gradient, and hence knowing the right way to swim. So this ancient molecular invention is a fundamental component of the behavioral repertoire of bacterium. They have many of these types of receptors, and they use them to find food, they use them to find poisons and go away from them. It's a very versatile and wonderful system. Now, why don't we think about something a little bit different now, something a bit closer to us, which is how do we see a cat? And I'm going to tell you a story about the cat and the code breaker. Well, when you see a cat, of course, you see it with your eye, and that image impinges on your retina, whereupon that image is converted into electrical activity that goes out in the optic nerve. Now, all of that electrical activity is converted into an elementary digital code. And if it were that you saw a dog, you would get a different digital code in your nerve cells. And in fact, every sensation is encoded into a digital code. But what is that digital code? Well, I'm sure all of you who have done your basic neuroscience will recognize that the neural code is the sequence of action potentials that are conducting along a nerve. These are all or none digital events, and all of the information is to be found within the intervals between them, the time intervals between those elements of the code. This, of course, is transmitted from your sense organs along those nerves into your central nervous system, and then something miraculous happens. That code goes into your brain, and somehow you see a dog. If a different code goes in, the one that came from your eye, you will somehow see a cat. So the question becomes then, how does the brain crack this neural code? It's one of the most important questions in neuroscience. If you're going to do it manually, to break a code, you might do something like this and do a lot of hard work and work out how this code, this language here, this linear B code, what it means. But of course, there are other ways to do it, and that is to invent machines that do so. And of course, you will recognize these important code-breaking machines from the World War II. But how does the brain do it? Well, the brain has a marvelous little code breaker in it, which is the synapse. Because the patterns of activity come into the synapse, that digital code, that neural code, it releases neurotransmitter and activates the post-synaptic terminal. And that post-synaptic terminal has some kind of magic going on inside it, which reads that code and decodes it. Let me show you how that works. Well, I'm going to describe for you now how synapses can read time in the neural code. But let's look at the very most elementary unit of coded information, which is this spike or action potential in the presynaptic terminal, the all or none event. As a result of the release of neurotransmitter, it produces this pulse, a pulse of depolarization of the postsynaptic terminal. That will ultimately drive that cell to fire. And it's the amplitude of that pulse that is not fixed, is in fact analog. In fact, the synapse is a digital to analog converter. And it's a very clever thing because it doesn't just do the same thing every time. If there's an interval between these two action potentials, that postsynaptic response will vary in amplitude. In other words, it's measuring the time interval and varying the amplitude accordingly. And if you put in a rather more complex train of activity, where there's 4 spikes, another interval, and so on, you see here that we get this variation in these postsynaptic responses, showing you that the synapse is constantly reading and modulating this output and generating this output from the code. So how might it do that? Now this is where the molecular biology comes into that. And about almost 30 years ago now, when the molecular biology of the synapse was really just beginning, it was thought that the synapse was a pretty simple thing. It was just made of, it was just a connector. And inside this postsynaptic machinery, there was only just a handful of proteins that could do all of that job. How wrong they were, because molecular biology and proteome technology shows you that these postsynaptic terminals of synapses, every one of those can house more than 100,000 to 200,000 individual proteins encoded by over 1,000 different genes. It is remarkably molecularly complex. And of course, in your brain, you have 1,000,000 billion of these synapses. So why is it so complex? Because you don't need to have all of those molecules just to do synaptic transmission. Those molecules are computing the neural code. And I'm going to describe for you how they do that. So into this synapse comes a pattern of neural activity, and there are different combinations of those proteins that read the intervals between these two action potentials. Another combination of proteins will read these the intervals between this one, and so on and so on. And that is how it produces this modulation. There is a combinatorial use of those proteins. Well, how do I know that? Well, here's some data from some of our experiments where we're looking at about 60 different genes here. Each one has been mutated and taken out of a mouse. And the synaptic electrophysiology has been measured in a part of the hippocampus. And we're presenting those different patterns or neural codes into each one of those mutants. And this color-coded matrix here shows you which genes are controlling which temporal aspect of reading that neural code. And if you look at this first point here, you'll see this particular component. There's a set of genes that control that. The red and the blue means increasing and amplifying and attenuating. And you have, for every different aspect of the neural code, a different combination of these postsynaptic proteins that are controlling that. And when you look at those combinations, what you find is that those combinations are in these molecular machines that are inside the synapses. So I'm going to explain to you how they're built now. Remember I said there were so many of those different molecules? Well, the genome is encoding large numbers of RNA molecules, the transcripts, which make all of the individual proteins. But all of those individual proteins are assembled in a combinatorial fashion to make these higher order molecular machines, which are then placed into the synapse, and they are reading the neural code. And those are the direct descendants of these ancient machinery that was measuring time in bacterium, which was used to find its lunch. that same molecular machinery, which is now very much more complicated, there's many more of them, they're much more larger and more sophisticated, those molecular machines are the ones that are decoding time and the neural code in all vertebrate organisms. And if you look at a bit more detail into the evolutionary history of these molecules, we start with these very ancient simple complexes of proteins, and in unicellular eukaryotes they became more complicated with the addition of more molecules, and they could therefore have greater functionality. And in fact, all synapse proteins, remarkably, all synapse proteins in the brain of every animal, all of those proteins that do all those extraordinary things, including controlling intelligence, all evolved before the brain. They all evolved in ancient unicellular organisms. It was all invented then. All that happened was that this molecular machinery was then co-opted and placed into these synaptic junctions when animals became multicellular. And it is this machinery that gives you all of these extraordinary capacity of transmitting information and decoding the information in synapses. But you'll notice a little diagram here. Here's some of our invertebrates which have these molecular machines, but you'll notice that they're much more complicated here in the vertebrate species. And if you look carefully at this, you'll notice that there must have been something very important happening within a rather short space of time. And this is what we refer to as the synapse complexity expansion, where there was a great increase in the number of those molecules. This is an intriguing and important event. And we can answer the question as to when it occurred. Because if you look at all of these different vertebrate species here, they all have these highly complex synapse proteomes, and you'll notice the times at which they have branched since the origins of vertebrates. And because they all have this highly conserved molecular machinery, you can deduce, therefore, that there must have been some molecular event that happened very early in vertebrate evolution that produced this marvelous complexity that we find that characterizes the vertebrate synapse. And this must have happened somewhere back in and around the Cambrian period. And as you can see in this nice picture here, there are many, many different types of invertebrate species that populated the planet at that time. And these are extremely sophisticated organisms. I would put it to you that these are extremely intelligent organisms capable of so many different aspects of behavior, just as invertebrates are today. But around this time, there was an organism like this one here. A fossil example might be that of the Epigenetics. An extant relative of that today might be the lancelot or Amphioxus. But it was in that type of organism that one of the most remarkable biological events occurred. And I'm about to tell you something that, if you do not know about this, should be probably one of the main top 10 pieces of biology that I believe everybody should know today. And what happened was this, that there was an ancient organism with a genome, and it was an invertebrate type organism, but one of its offspring had, by virtue of a genetic accident, the largest mutation of them all, an entire genome duplication, just one of its offspring. And miraculously, that offspring, sometime later, for those of you interested in detail, approximately 50 million years, there was a further offspring from one of these organisms that had a further duplication, hence multiplying by 2 and 2 again to produce 4 equivalent whole genome duplications. Now, why might you say that is so interesting and so important? Well, the remarkable fact is that it was from that organism that every vertebrate on the planet today descended. Had that organism died, there's no guarantee that any of us would be here today. Think about that if you're interested in life and intelligence in the universe. This is a very rare and very special event. Now, what this does is, and it's a diagram here, if here's the present day and we go back to the first and second genome duplications, if there's a single ancient gene of the kind that you might find in an invertebrate species, and here's a linear diagram of a protein encoded by a single gene in an invertebrate organism, after the first genome duplication you get 2 copies, after the second duplication you get 4 copies, and as you can see, all of these copies, which you will find in the genome of humans and other vertebrates today, you'll see they're all very, very similar. In other words, they've just varied a little bit by accumulating mutations and variants over that time period. And it was this molecular duplication type events that increased the molecular complexity of this post-synaptic machinery. And if you're interested in experiments that demonstrate that that expansion in genome complexity gave more complex behavioral repertoire, more sophisticated components of the behavior, I would point you toward these manuscripts down here. So that's all very nice and all very interesting to see that this molecular complexity has expanded. But how do you get a complex representation out of a neural code, a complex representation being something like this type of image here? Surely one synapse can't do that. Well, I'd like you to look carefully at this picture. And this is a rather spectacular and beautiful picture, because what you're looking at here is a view of a brain section taken from a mouse, and each one of those little dots are excitatory synapses. But you'll notice that they're all a bit different. They are color-coded because they have molecular and physical differences between, amongst them. And you can see there's all sorts of interesting patterns. It almost looks like sort of, this is a cell body layer, the CA1 region, his dentate gyrus, and you can see beautiful architecture and diversity and distribution of an extraordinary diversity of synapses. Generally, we've been thinking that the nervous system has just got excitatory synapses, and all excitatory synapses are made of the same stuff. Well, they're not. They're actually extremely different. And now I'm going to explain to you how this synapse protein complexity is related to synapse diversity. And it's remarkably simple. Those genes which make those proteins, which make those molecular machines that are doing that decoding job, are not all just placed into each synapse equally. They are distributed such that some of them go into this type of synapse and some of them go into another type, and then you get others with various mixtures of those. And because there's many of these types of machines, you can get a great diversity of different types of synapses, which of course have different physiological properties. It's amazing to think how many there might be. And this is just a number, we've done some analysis. Even with two proteins, two of these postsynaptic proteins, you can generate 37 synapse subtypes. With 10 proteins, you could produce 10 to the 11 numbers of synapses, which happens to be the total number of synapses in a mouse brain, only with 10 proteins. But those of you who've been paying attention will know that there's more than 1,000 proteins in individual synapses. And so it's quite possible that no two synapses in the brain are the same ever at any one point at all in the lifespan. And this is possibly an underestimate because there's many other sorts of molecular differences in these proteins that again could produce even further complexity. So our brain isn't just made up of simple excitatory or inhibitory synapses. It is in fact made up of a vast library of different types of synapses. Now let's ask ourselves the question, what might that do at a functional level? And here's a little diagram of two nerve cells, and you have the dendrites emanating from those two nerve cells here. And here's an axon that's coming in and touching both of those. And this axon is intersecting with the first cell, and you can see that I'm calling that synapse A, and it has a particular combination of proteins. And over here is synapse B on the second cell, and it has a slightly different combination of proteins. And you'll remember that I showed you from those genetic experiments that depending on the proteins that you have will determine how you respond to a pattern of activity. And I'm showing that in a very simple way here. Here is a little pattern of activity, a little neural code of 7 action potentials. And here are these analog postsynaptic responses in synapse A. And here they are in B, and you'll notice there's a different pattern. In other words, the molecular composition of these different synapses will determine the physiological response of those synapses in the spatial fashion. Here's a really simple way to look at it. I've got 36 synapses here, and I'm going to just call them two different types, these sort of dark ones and these light ones here. They have different sets of proteins that are in them. And this is type A and type B. And we're going to stimulate them with a pattern, all of them equally, with a pattern of neural activity. And I've drawn it in such a fashion that these ones on the outside had a different response to those in the middle. But these synapses can respond to any pattern of activity, and some patterns of activity, such as this one, will come along, and this pattern of activity will not produce a differential response because those two synapses can't tell the difference between them. And in this way, I hope you can see that different patterns of activity can come into a map of synapse diversity and produce differential representations or functional outputs. Let me show you that in another way. We have a little map here of about 11 by 11 synapses, and they're made up of these different molecular compositions. And if an animal is sensing or experiencing something in its environment, this pattern of activity will be produced. And this is the response of that map. And you'll see sort of zones and patterns light up. And this is just a sort of a heat map representation. Now a different pattern, a different behavior will produce a different neural code. And this will drive this synaptic map or synaptome map to produce a different physiological output. And a different pattern again will produce a different output, and so on and so forth. And it is as though when you're, all of these activities produce a different response from the same synaptic map. And therefore, if you think about this as a sequence of behavior that the animal is going through, it's kind of a mental movie where those synapses are changing in their activity pattern as the animal is experiencing it. And that is because those synapses are constantly breaking the neural code and doing so in a spatially organized fashion. And this produces internal representations in the brain. Humans have these maps too. And in this biochemical study of a variety of different areas of the human neocortex, we characterized in great depth these thousands of proteins. And in all of these different areas here, there's differential protein compositions corresponding to the fact that there are different synapse types being distributed in those different parts of the neocortex. And this is just one of these single synapse resolution maps that we've been generating of the human brain. In fact, there's many aspects of these maps in mice and humans that are very, very similar. Now, why might these maps be interesting with respect to human neocortical function? Well, I've just explained for you how the mouse will, when it senses different experiences or different inputs, will use these maps in a differential fashion. And we, of course, are interested to ask the question, could that have anything to do with the way, when one looks into brain imaging, functional magnetic resonance imaging, for example, does that in any way have a relationship between the synapse composition in those different parts of the brain and the responses that are seen in this sort of tasks? And if one takes functional MRI data from the Human Connectome project, where there's many different particular tasks that generate functional responses in the brain, such as these ones here, language tasks, math, emotion, face recognition, and so on, that you can find that there are different compositions. We can relate the protein compositions in the parts of the brain to these various behavioral tasks and show that there is certain tasks map on to different synapses in different areas of the brain with those different compositions, consistent with the type of stuff we see in our basic science at the level of the mouse. So these synapses and these proteins are obviously of great interest with respect to many aspects of human cognitive function. This is a meeting on intelligence. And I'd just like to mention that a recent paper that came out, a very large genetic study on intelligence, points directly at these sets of proteins that I've talked about today. And in other projects with my own group and Ian Deary and Tim Bates and others, we have also shown in the past that these proteins that are doing all of those wonderful things that I've just shown you are in fact involved with human variation in intelligence. But there's a flip side to all of this. And the flip side, of course, is that those molecules, which might be doing all of these wonderful things here, might also play some sort of role in various sorts of mental disorders of one kind or another. And this is just a typical example of a small family in which one of the offspring, in this case Edouard here, spent his life in an institution in Switzerland because he had what was almost certainly schizophrenia. And of course his father, Albert Einstein here, certainly didn't suffer from that. And these days we know that many of these sorts of families of this kind have what's known as de novo mutations in various genes that are important in the nervous system. And if you were to ask the simple question now, of all of those very interesting proteins that are in the post-synaptic terminals of excitatory synapses, what is it that they might have to do with brain disease? Well, a number of years ago when we were studying the human synapse material, we recognized and identified over 130 brain diseases that are caused by mutations in over 200, it's now over 300 genes that encode postsynaptic proteins. And there's many, many different sorts of disorders, including these common disorders, schizophrenia, autism, intellectual disability, and dementias. So I think it's true to say that at this time, those postsynaptic proteins carry more a burden of disease in the nervous system than any other set of proteins that we presently know. Now, one of the most interesting aspects of this, or one of the most puzzling aspects, is, in my view, is this question of schizophrenia. And indeed, again, in a number of papers, including these ones here, in which we were involved, they used our molecular data sets to find that there was an increased burden of mutations associated with schizophrenia in these postsynaptic, in these postsynaptic proteins. And that leads me on to talk about something which I consider to be a really terrific puzzle. And here's the puzzle. Schizophrenia is the archetypal adult onset disorder. So why, if you have mutations, do you have to wait 25 years for it to come on? And I'd like to just come back to that a little bit later, but I'd like to tell you about some insights that have come from, again, basic science type of approach. And this is a small study that we published last year where we were interested in the changes that might occur at the molecular level in the brain across the lifespan. But let's place that into a little bit of context just for a moment. I found this, and I'm sure in this audience many of you will be quite familiar with the Reminiscence Bump. Of course, everybody likes to think that their life in their 20s and young period of time was all that exciting and all that interesting, and maybe that's why you tend to remember it more than things in the years after that. But it could be that there's some interesting biology going on there. And I believe fluid intelligence is also sort of peaks around about the mid-20s and early 30s, and there is documentation of creativity as well. So it's clear that it's a very interesting time in the brain over the lifespan. But most striking of all is the fact that 75% of all mental disorders have an onset in adolescents and young adults. And this, of course, is a tremendous personal and social and economic problem. And as I've already told you, schizophrenia is really a prototype such disorder. So let's talk about molecular biology here. So here is the birth until the year 80 in humans. And we're simply going to ask the question, how much does the level of any gene expression level change across the lifespan? And if one is to measure lots and lots and lots of time points here, you can actually plot a trajectory. Trajectories are interesting because when a trajectory turns, that is because there is a regulatory event. You're turning the steering wheel. And in this case, it's a gene regulatory event. And in a side project, I would call it, in the lab, with one of my graduate students, Nathan Skeen, we did the following experiment. We thought, we'll have a look at this in the mouse. We had a lot of mice, 186 of them, aged across a wide range and scattered, and we took out the RNA from their hippocampus, and for every age point, we measured every expressed gene, and therefore for every gene, we could plot the trajectory across the lifespan. And for every one of those trajectories, we identified these turning points that occur. And we could then identify patterns in this gigantic set of data using machine learning approaches. And we came up with algorithms that allowed us to test if they had any predictive value. And here's a kind of prediction that's pretty interesting. We can ask the question, can brain age be predicted from an RNA sample? So here is the x-axis showing the actual age of the mice. And here's going to be the predicted age. And here's how we do it. We take an RNA sample that is not used to generate these predictions. And we ask the computer, how old does it think the mouse is? And that's what you find. That's a pretty nice graph. And it's a rather important, striking graph in my view, because it tells you two very simple facts. The first is that you can tell the age of a mouse by looking at the level of gene expression in the brain, and moreover, you can do it across the entire lifespan, which tells you then that your brain is never the same twice. It's always changing. You might say, well, that's all very interesting. Does that same thing happen in humans? And in fact, we did the same type of experiments using human gene expression data from 14th gestational week to 78 years. And that's what we see, the same thing. Bit messier, presumably because of post-mortem effects, and it's not ideal in the laboratory, but it's the same thing. And if you wish to read much more about the details of this, I refer you to our eLife paper last year, because we have analysed the molecular details of these trajectories across the lifespan, and they are the same types of trajectory in a mouse, which only lives for 2 years or less, and a human, which lives up to 80 years. And it's the same genetic program that is going on in both species. What this tells us then is that at every age, your brain gene expression is different, and it's like turning the pages in a book. As you get older, every year, every month, perhaps even every week, there are changes. And I consider it to be like a sort of a genetic biography across the lifespan. And we refer to this genetic program that produces these changes as the genetic lifespan calendar. Now, this calendar is a pretty interesting thing, and we thought we'd like to ask a few more questions about it. Here's a really obvious one. When do most genes turn? Would anybody like to hazard a guess?
17.
Well, here's the graph. Can you see the peak? Males 26, females 27. Highly statistically significant and statistically significantly different. So here's just another way to look at that, a cumulative plot here, and see lots of changes in this young adulthood that are going on here. By the way, I look around the room and I feel I put myself in the same category. I'm up in this rather boring category up here. This is after midlife. Notice how there's not much happening genetically in your brain in terms of gene regulation. All the excitement is when you're a young person in your 20s. So what is that excitement? What is actually going on? What are the genes? And of course, we know all the genes, and we can ask which ones are they? What sort of things do they do? And then we plug all of that in and simply ask what sort of proteins do they encode? And this is what we find. We find, miraculously, it's our old friends, the synaptic proteins and the post-synaptic molecular machines. And that tells you that in the young adult brain, that molecular machinery is undergoing some pretty significant changes. We could also do more with this set of data. We could look at that young adult regulated set of genes, and we could ask in what way is it relevant to gene sets that are involved with various sorts of disorders. And we found that those sets of genes are highly enriched in susceptibility genes for schizophrenia. Now, I think I've already set you up for this, but schizophrenia, of course, has a young adult onset. It also has a female delay that is about 2 years, and this is precisely what is seen in this data set. And what this suggests to us is that there must be molecular changes that are going on in the young adult brain that are changing this molecular organization of these synaptic maps in the brain. And we believe that it's being normally reconfigured in the young adult brain. And if there are gene mutations, then that reconfiguration process does not work properly. And so we have this sort of genetic calendar model of schizophrenia, which is this one here, which is that the genetic susceptibility of schizophrenia remains silent until young adulthood. The mutations are not showing any effect, but it only becomes exposed by the gene regulatory events in the genetic lifespan calendar. So in effect then, schizophrenia is caused by mutations that target an age window in this genetic lifespan calendar. And this coincides with this peak age of synapse genes that are reorganizing. Quite different hypothesis to the standard one, which is that there is some sort of insult that occurs during fetal life, and then a second insult that happens in your young adult period. You smoke too much pot, you have the wrong parents or something. But in fact, we're putting here, as you can see from this molecular data, a very strong genetic element to it, not only in the origins of the mutations, which give the susceptibility, but in the genetic program that controls when those genes are expressed across the lifespan. So I'm going to conclude. We see these molecular data molecular organizational principles of how these proteins are encoded and built into these molecular machines and placed into synapses and put into what's called synaptome maps as some kind of Rosetta Stone, which allow us to look at lots of different problems which I've shown you today. And I hope I've been able to convey for you some of the interesting aspects of this molecular model by looking at its origins and ancestry, going back to the very earliest forms of unicellular life, and how that molecular machinery is responsible for these animals to survive and find their lunch, and how this sort of machinery is involved with the perceptions of complex images and the decoding mechanisms at the level of individual synapses, and how through these dramatic expansions that we have seen in this sort of Cambrian period did we see the origins of these very much more complex nervous systems with their very sophisticated maps that we now find distributed across our nervous systems in the brain, and the relevance to aging. So I'd like to leave it there and thank you very much for your attention, and leave this slide of acknowledgements of many of my colleagues. Thank you very much. Take some questions? Yeah, happy to take any questions.
Sorry. One of the concepts that's evolving in intelligence research is the idea that some brains are more efficient than others. And this has been studied with the connectome data and look at the efficiency of various brain connections. And those connections have been used to predict IQ scores. I'm wondering if there are quantitative ways to look at how efficient some of these types of synapses might be, and whether the types of synapses can be clustered into groups that would be meaningful for predicting something like an IQ score.
Let's talk about efficiency first. The synapse converts the digital to analog information, and it reads different sequences of activity. And every different synapse will give a differential response. Now, you might call that efficiency. In that, synapse A will give a larger response to a particular neural code than synapse B. In other words, there is a quantitative difference. I wouldn't call that efficiency, I would just call that a differential response. You could equate it to efficiency in some way. Now, if it is that there are certain patterns, if I have a population of synapses over here and a different population of synapses over here, this population over here might be particularly good at responding to certain patterns of activity. Perhaps you'd call it more efficient. They could be patterns of activity associated with language. These ones over here might be particularly good for responding to visual inputs or patterns associated with those, and you might describe those as being particularly efficient for sort of visual aspects. So the differential distribution of synapse types into populations could meet some of the criterion that you have described.
I'd be more interested in if you have two individuals, person one and person two, whether they could be whether something about their synapse maps could be quantified. They would have the same types of synapses.
Right. So let's just say now I have a synapse map, which I do. As a result of genetic variation, each one of us in this audience will have a slightly different synaptome map. Synaptome is the word, by the way. Synaptome refers, like genome, gene is to genome as synapse is to synaptome. It's the full complement of synapses in the brain. And the synaptome map of each of us is a bit different as a result of our genetic variation. So there's no question about that. And so you could just simply impose those kinds of genetics onto that. There are a lot of interesting questions about how genetics change synaptome maps. And what I didn't describe for you here, but will be shortly published in the journal Neuron, is evidence that gene mutations reorganize, or what we refer to as reprogram, the synaptome map. And so it's very clear that different mutations alter the synaptome map. In fact, our evidence supports in this paper that we think that every synaptic protein, and possibly many other neuronal proteins, gene mutations, will change synaptome maps. We think it's going to be a general property of changing the nervous system. And so, of course, normal genetic variation, whether it be strain variation, individual variation, will have produced variation in synaptome maps as well. I think in terms of measuring synaptome maps, as I've indicated for you here in human studies, obviously in mice we can do it with great detail, and obviously in human postmortem material we can do it in great detail. But at the level of doing it in the intact living human, the approach that we're interested in doing is to apply molecular markers that are compatible with various imaging modalities that allow us to quantify different synapses and synaptic types. That sort of work is in its very early stages, and it would appear to be possible in principle. And in the first case, that type of technology is now being actively pursued for studies of mental disorders, brain injury, schizophrenia, dementias, and all the rest of it, which we already know to change the synapse map. Yeah.
Interested in the implications of your research for diseases like schizophrenia. So schizophrenia, for example, has been pretty impervious to treatment. One of the main treatments is dopaminergic antagonists and environmental manipulation. So if it would seem to imply, your data would seem to imply that the only effective treatment for schizophrenia would be gene editing.
No, I wouldn't imply that.
Okay, what would you suggest would be the cure for schizophrenia based on your research?
I think you've got to look at it in two different ways. One way is that schizophrenia and many other disorders will be associated with a change in the synapse diversity and synaptome map. So in a very simple-minded way of thinking, you might ask, can I now modify a synaptome map with some exogenous thing, a drug perhaps? Who knows what other sorts of treatments and modalities could be applied. And until one tests all of those different modalities, we do not know. But we do know from some work that is as yet unpublished is that we can use various drug treatments, including olanzapine, to modify the synaptome map, at least in the mouse. So that's an example of some sort of pharmacological manipulation. But I'd like to step up to something slightly bigger and more interesting here. I mean, one of the great dilemmas of the genetics of schizophrenia is the fact that there are so many different genes involved. And if one wants to think about precision medicine, which in its simplistic form could be reduced to a statement like this, which is that for every mutation I have to have a special drug, then you're never going to make 250 special drugs for 250 genes involved with schizophrenia. This is not going to happen. So what's the alternative? Well, there is an alternative that's suggested by the data that I've shown you here today, because what we have shown you here is that really independent of the genetic mutation, that the onset is governed by this genetic lifespan calendar. And if it were that you could modify the genetic lifespan calendar, you might be able to abrogate in some fashion, to some extent perhaps, the penetrance of the mutation in the young adult brain. So one might be able to think of a whole different avenue of approaches which are aimed at modifying the genetic lifespan calendar and test if you can change that and see if you can then prevent mutations. That has the advantage that you might be able to do that with one drug, which would be a blockbuster.
Yeah. Do you think you have an avenue to slow or stop the genetic lifespan calendar?
There's no reason to. There certainly will be genes that will do that. It would defy biology and genetics if there were not such genes. They need to be found, and research needs to be done on that.
Hello, and thank you for your wonderful presentation. Do you think, based on your study, there might be a common pattern or anything common in terms of like disorders such as schizophrenia in synaptomatic map, or no?
Yes. If I've understood you correctly, you're asking about the similarity between disorders? Yeah. I have no doubt that if one could generate these synaptic maps or synaptome maps in humans or any other organism, one would be able to compare and contrast them with one another. We have, as I mentioned already, have done some disease studies using mouse models of diseases, which have mice carrying gene mutations that are involved with different disorders. And we're seeing these beautiful changes in different maps across the nervous system in every single one of these mice that we look at, every one of these different mouse disorders. We have also looked to ask the question, is there any relationship between these maps and the cognitive functions which we have measured in these mice? Several years ago, we published analyses of mice carrying mutations in proteins, and these mice were studied in touch screens with various aspects of higher cognitive function and attentional tasks. And we also tested some humans in the same tasks as well. And these mice that carried these different mutations had a different sort of profile of cognitive impairments. And we have found that there are some of these, we've measured the synaptome maps in some of those same mice, and there are some rather nice sort of dichotomies and parallels between some of those changes, which one would, I think, expect at some level. So I think there will be ways to quantify these maps and relate them to different cognitive functions and different regions of the brain and classify diseases in new ways.
And do you think that could be a solution for the autism spectrum disorder later once they found, a more common pattern of these, synaptologic maps?
I wouldn't use the word solution. What I would say would be at this stage, what we need to do is to do mapping across the lifespan. Just as I said for that gene expression stuff, one wants to make these synaptome maps. I didn't show you the actual data on these synaptome maps in detail. As I mentioned, it'll be published in Neuron in a few weeks from now. But we've made the first whole brain synaptome maps of any organism. And for the first time, we can quantify and look at the molecular distribution of individual synapses, looking at about 1 billion individual synapses in the mouse brain. In an ongoing project, we have been doing that across the entire lifespan of the mouse. And what I can tell you about that is that at every age, your synapse maps of the brain are different. And you can tell the age of an animal by looking at a synapse map. And therefore, one wants to ask questions about autism mutations, schizophrenia mutations, whatever mutation, and see at what age they produce their changes in those sorts of maps. And that's ongoing research at the moment.
Thank you so much.
Thank you. Any further questions? Yeah.
Enjoyed your talk. The question is, do we know possible reasons, or do you have a theory why molecular mechanisms at post-synaptic level peak at mid-20s and relatively stabilizes thereafter or declines? And if we did know that now or in the future, is it possible to manipulate that?
I think I've answered the second question already, which is the possibility to manipulate it. I think it will be possible to manipulate it. I see no reason at all to say why one would not be able to manipulate it, just based on what we know about gene regulation. But let me go back to the first question, which is sort of why do you see all those changes? Well, you see a lot of changes in the 20s, and one of the more interesting, I shouldn't say more interesting, one of the other interesting aspects of this is that by about 40, about 95% of these regulatory changes have occurred, and all the gene levels have sort of plateaued. And I didn't go into the details of this, but essentially what's happening in your 20s is that all these gene levels are changing, and then they typically plateau, and they reach a set point that you have to live with for the rest of your life. And so if you don't get that set point properly, you're going to have to carry the burden of that. So why might it be that there are these regulatory changes or that sort of stop happening after somewhere in the mid-30s or thereabouts? And similarly, when you look in the mice, why is it at around about 6 months of age in the mouse? Well, I think there's a very interesting observation that may be relevant to that, which is the fact that up until only a few thousand years ago, nobody ever lived beyond the age of 30. And there's very good evidence, and I've consulted with experts in paleontology about this and looking at all these tooth samples and so on. But effectively, everybody was dead by the age of 30. And so there's been natural selection acting up until that point of time. There hasn't been any natural selection on living beyond that age point in humans, and that's almost certainly true for mice too. They don't typically live for 2 years like they do in our lab. They only live for about 6 months or something like that in the wild. So if you were now to think of the normal lifespan of the organism, which is not what I've shown you today, I've shown you an extended lifespan, then you're seeing that throughout the normal lifespan, there's a lot of changes at every age. It's just that they slow down after this sort of extended lifespan period. And I would simply put it to you that that would be for setting up the normal daily contingencies, quotidian events that occur week to week, month to month throughout that very important, first 30 years of your life. So I think it would be a natural selection would have built this program in that way. Okay. Pardon? Yeah, I'll take a couple more questions, but I assume people have other things to do.
Thank you very much for a marvellous lecture, which I enjoyed immensely. I'm asking you to make a seditious view about intelligence, which is that most organisms seem to have the intelligence they required for their survival. And they did that at the synaptic level, if I can say that. their mental pictures were good enough to survive. So try and pick up the theme which some others have done. It's not efficiency, but it's power. Most of our work is about trying to distinguish why some people seem to have more powerful intellects than others. Is there some way in which, by looking at the synapse, one can give a very informed of the relative mental powers of different animals, different levels? Is there something which gives us the thing we're sort of after, which is a computational power measure?
Yeah, I think that's not too difficult. What I've shown you is that the molecular machines that you're using to watch my presentation and ask your question, those molecular machines are the same sorts of molecular machines that E. coli have. But I would put it to you that your question is much more interesting than one that would be placed by an E. coli. And the reason is because you have a lot more of those molecular machines, they're more diverse, and you've organized them in a spatially distributed fashion within your nervous system in a way that the E. coli cannot do. And so if you wanted to make some equation with power, computational capacity, I would put to you that one of the key elements here is diversity, and diversity of synapses and diversity, and therefore diversity of responses.
So I may be 10 E. coli.
No, you're probably many more than that, actually, and you can do an estimate. That genome duplication has done wonders for you. Any further questions?
Just one other concept that you hear a lot of talk about is epigenetics. And this concept didn't appear anywhere in your lecture, as far as I could tell. Do you think that's an overrated concept, in that the genetic story seems to unfold more or less independently of any external events, or even something general like stress?
Dobzhansky said nothing makes sense except in the light of evolution. And if you look at epigenetics, DNA modification by methylation and other sorts of mechanisms, they're recent events. They only came about a few 100 million years ago. And I would place it, therefore, in the category of many other types of physiology and processes that have arisen in the last few 100 million years. They're typically modifications or modulators of more ancient and more fundamental and ultimately more important machinery. I wouldn't try to rate them with respect to one another, except to say that the most ancient and most conserved machinery, in my view, is almost certainly the most important stuff. Okay.
Thanks again for
Right. Thank you very much. Yep.